Hi everyone, my name is Xiaoman Li. I will have a spotlight talk about the report called Design of Specific Primer Sites for SARS-CoV-2 Variants Use the Evolutionary Algorithm. The detailed information about the report and author could be checked in the video information below. Maybe you would be unfamiliar with the SARS-CoV-2, but you must know its member COVID-19. It should be the most popular topic today, where more than 100 million people was infected since last year. And a more serious thing is that there are 1 to 2 new mutations or we call it variations of the SARS-CoV-2 every month. Moreover, part of these variations may not be detected by the primer designed for the SARS-CoV-2. So what is a primer? Basically, it could be used to test the virus, and it's a short RNA sequence with 4 kinds of the nucleotides. Just like the graph shown below, the work of the design, the primer could be regarded as the distribution of these nucleotides. In this report, the author aims to introduce an evolutionary-based approach to find good primers candidates, which is more efficient than the traditional way. Let's look at the primer design. Just like I mentioned before, it could be used for COVID-19 test. But for the new variants, there should be the new primer be designed. And how could the primer work? So the graph in the right side briefly shows the process of the test, which is also known as the PCR reaction. But no worries, it's not necessary to know how it works in detail. We just need to know the primer will influence the result of the test. The primer, which is a 18 to 22 sequence, which could bind to the viral RNA, and if so, there will be the fluorescence signal to show the positive result. Usually, there are some software based on the deep learning used for the primer design, such as the Primer 3 Plus. Before we talk about the evolutionary algorithm, I will introduce that what is the features of a good primer candidate. As you know, it will influence the fitness function of the EA. Firstly, the length of the primer should be between 18 and 22. Maybe we could just set the length when we design the individual. Secondly, the melting temperature should be close to 60 degrees. Then the C and G nucleotides should account for about 40 to 60% of the sequence. There are more factors but may not be considered in the fitness function. For the traditional solution based on the convolutional neural network, there might be some drawbacks like the low efficiency and high discard rate. Maybe everyone has know the basic parts of the EA, so we just start the introduction. To the evolutionary algorithm, the first step is to know what is the individual and how to initialize the population. I said that the effective primer's length should be 18 to 22. So in this report, the test used 21 as the length of the individual, and here is an example for the candidate. Then, after we set up the individual, we could initialize the population. The author used the size of 100 for the first generation, and for each of the offspring generation, the size is also 100. I will also introduce the replacement of the population later. The critical part of the algorithm here might be the fitness function, different from some typical NP-hard problem like the class P. The fitness will only be influenced by one factor, which is the distance. The fitness here could be calculated by the weighted sum. As the function here shows, there are 4 parts includes the function pi to check whether the candidate primer inside training sample, and ci represents the percentage of the GC base of the candidate primer, and the ni consider the presence of the n symbols in the sequence, which indicate an error in the rate. The ideal primer candidate should only contain the A, C, G, and T values for the RNA. The last one, Ti, is used to indicate whether the melting temperature is close to 60 degrees or not. Overall, these four functions represent four key features of the better primer candidates, and according to the importance of each features, the author also introduced the weight for four functions in this report, which are 1, 100, 1000, and 1. The next step are to identify the selection, crossover, and mutation. For selection, the author used the tournament selection here with the selection size of 2, and for each generation, the entire population will be replaced according to the fitness. For the crossover, 1 point crossover was used, and the probability of the crossover PC was set to 0.85. As for the mutation, the author used a biological principle here, but doesn't matter. It can act on the two integer value k that represent the index of the sample in the training site, and the position p, which represents the position inside the sample k's genome. Basically, in the mutation of the p follows the normal distribution between 1 and n, which was done to ensure that the large probability of the p was close to the half of the n, and the small probability was on the left and right side of the interval. Because the changed value of fitness function will be smaller on both two sides, probably we choose the mutation rate as 0.15, and these two tables shows the test results of the application of the evolutionary algorithm in primer design after 20 runs. There are 6 of the best primer candidates. As you see, although all these sequences in the left side is with a high fitness, but there might be still some reasons so that it cannot be used. The author mentioned that it only costs 62 minutes for one run, which is much less than 16 hours of the traditional way. The generated forward primer of B117 variant appeared in 95% of the sequence. If we only consider the B117 variant sequence with more than 5 mutations, so the frequency of the appearance could up to the 99%, which is really high. And then the application of the evolutionary algorithm in primer design should be significant research. Firstly, the primers found with evolutionary algorithm are exclusive to the particular test variant, such as the B117. It means the primer is reliable to test a target variant. It figures out the critical issues of the design the primer for variations. Secondly, the primer designed by EA could accurately diagnose new emerging SARS-CoV-2 variants. And currently, diagnosing and monitoring the variants will be the critical important thing to help fight with the spread of the virus and help the government to develop new policies. Certainly, just like the author said, the research until now is still in silico validation, which means the next step in the progress will be to test the primers in the lab. And comparing with the traditional method, there are some typical advantages of the EA, such as higher efficiency, which could generate qualified primer, and the fitness function could be able to include the constraints so that they decrease the unsuitable primer candidates. For the traditional deep learning approach, it uses a platform called Primer 3 Plus. Takes about 16 hours to find a suitable primer for each variant. It cannot generate enough primers candidates because most of them are discarded. It caused by the unsuitability, like the low presence of the GC bases or the higher or lower melting temperature. In my viewpoints, There are both advantages and some potential issues of this research. For the positive aspect, the author used the evolutionary algorithm to automate the traditionally time-consuming primer generation process, and design the weighted fitness function to formulate the constraints on the primary candidates. But what's more, they also compare the results between the EA and the traditional DL method. However, I think there are still some potential chances to improve some point. For example, I don't think only a weighted fitness function is inclusional way for formulate the constraints, and there is no guide for us to set this the weight parameters like 1, 10, or 1000 because this number are just designed only for introduce the bias among 4 criteria. It's more likely to model it as an optimization problem, I think. And I think multiple mutation and crossover implementation could be provided, and the parameters are set for combination. A cross-validation mechanism could also be introduced to get a higher level automation, I think. Overall, the topic of the research really attracted me because COVID-19 might be the most serious challenge faced by the human being today. The application of the evolutionary algorithm to design of the specific primer should be the good research direction in the future, I think. Thanks for watching.